Okay, happy Thursday, everybody. Welcome to our bi-weekly IMAG-MSM working group on multiscale modeling and viral pandemics mini seminars that happen every other week at this time. Please note, as always, the webinar is live streamed to YouTube and is also recorded. And this gives us the opportunity to also share the presentations with the community in case they can't make it at this time. So just a couple of housekeeping items. So as always, please feel free to contact James or I or Jim Sluka with any questions. Lorenzo as well. Lorenzo has been tremendous help in helping us organize the webinars every other week. Of course, we have a few social media presence, so please feel free to post content relevant to the group or the Global Alliance for Immune Prediction and Intervention that was recently founded as a build-on, as an extension of this multiscale working group. Please follow us on X. Again, feel free to share any content that might be relevant to the community. A few announcements you may have seen or may not. Last week, or maybe this week, I think it was earlier this week or late last week, NIH announced the new funding opportunity that is very relevant to this community. U54 Centers of Excellence for Systems Modeling of Infection and Immunity across biological scales. Note that they require that one of the projects be HIV related. The deadline for the submission of this competition is June 30th of 2024. There are a couple of faculty positions available in Indiana University. Please feel free to contact James Glazier. He will have also additional information if anybody's interested. And the University of Florida is also looking for faculty positions in the Computational Systems Medicine, the Laboratory for Systems Medicine. Again, if there are any questions, you can contact Reinhard, who will also have potentially additional information. Otherwise, you can find these positions on these links. Recently, Reinhard had published a paper that discusses the meeting at Lake Nona. That was one of the first medical digital twin meetings last year. So feel free to check that out. It's another example of this community coming together and publishing papers and working together. If you have any publications that are relevant to the community, please feel free to email them to Jim Sluka at jsluka@iu.edu, and we will promote them, advertise them on our website. Are there any other announcements? And if you have any announcements that you find throughout the 2 weeks prior to the meeting, please feel free to send them to us, and we can include them in on the slides here, like we're doing during every webinar. There will be no steering committee tomorrow, as James and I are both traveling. So there will be the next steering committee meeting will be in 2 weeks. The next webinar will be on April 4th by Rebecca Morrison from University of Colorado on models calibration, validation, and uncertainty quantification. We don't have a speaker currently scheduled for April 18th. If you have any recommendations, would like to nominate somebody or nominate yourself, please do so. Email us, and we can add you to April 18th. And of course, if you have any other nominations for, down the road, please let us know. You can suggest speakers at this URL, or if it's easier, feel free to email James, Jim Sluka, Lorenzo, myself. Be happy to add you or anybody that you would suggest that would be relevant. It's my pleasure for today's presentation to introduce Hugo Geerts from Certara, who will talk about, who will share with us their information about their Neuroplatform.
Okay, so first, thank you to Tom and James and folks at Indiana University for hosting this webinar, which will be actually talking about a Neuroplatform, which is a kind of a comprehensive mechanistic quantitative systems pharmacology model for Alzheimer's disease. And I want like to stress the term pharmacology, because we're going to talk about drug and therapeutic interventions on top of disease models. So just as a kind of a general background for the people that are not really working in the Alzheimer's disease field. Okay, got it. Let me to my pointer here. So Alzheimer's disease is a public health concern, because more than 55 million people are diagnosed with dementia in the world right now. And the cost is staggering, both the direct cost for treating those patients, but also the indirect cost about the burnout of the caregivers. And that will amount to about $500 billion a year, and soon will actually surpass $1 trillion a year. So it's a public health challenge to try to treat all those patients. There are only a few treatment options that are approved and on the market, starting all the way in 1992 with tacrine, and then a couple of acetylcholinesterase inhibitors. And I was lucky to be involved in galantamine to help that bring on the market. But you see that between 2004 and 2021, we had about 200 trial failures. So it was a kind of a very gloomy time period, until, of course, the amyloid antibodies came on board with aducanumab, lecanemab, and then maybe donanemab soon. Now, the idea is the following, that this is a kind of a cartoon showing the kind of the community consensus, more or less, about Alzheimer's disease. So there are a couple of major pathological hallmarks. One is the so-called beta amyloid plaques, which are extracellular plaques of a peptide called beta amyloid, which then are leading to what we call intraneuronal neurofibrillary tangles, which consist of the protein tau. And sometimes you might also see that dementia pugilistica, which is traumatic brain injury, might directly lead to tangles. And then those two together actually are supposed to lead to a kind of neuroinflammation, which are going completely awry and out of control. And you could actually compare that to the kind of beta amyloid being the match that sets the fire, the tau being the spreading bushfires, and then ultimately the neuroinflammation, which is the kind of the forest fire. So what we are dealing here, to go from the molecular biomarkers to underlying pathology to the clinical function in Alzheimer's disease. So there's a lot of studies done on gene-related causal networks, which leads to some of the understanding of the biology of the key landmarks, amyloid plaques, tangles, and microglia activation. And nowadays we have clinical biomarkers like PET imaging tracers that allow us to monitor the dynamics of amyloid and tau inside the living brain. We also have fluid biomarkers that give us an easy, accessible way into looking in what happens inside the brain. But the big challenge is to go from those pathological hallmarks to what happens at the level of the neuronal excitability and the neuronal functioning through the synapses here, ultimately leading to functional biomarkers like maybe EEG or cognitive tests that you can actually do with patients. And then finally leading to what we would call the clinical outcome, which is ultimately what the regulatory agencies want to see. So a positive effect on what they call an ADAS-Cog, which is a clinical scale that tests the kind of the cognitive performance of the patients, or a CDR sum of boxes. So the challenge is to go all the way from this particular pathology to the kind of the functional biomarkers. And so today I will show kind of a strategy to actually try to perform that. So we will talk about the concept of computational neuropharmacology, which is a way we think we could simulate functional clinical scales starting from the underlying pathology. And then we will actually go back to the amyloid aggregation modeling, the tau pathology modeling, and ultimately ending up with virtual twins. I noticed in your introduction that you are very fond of virtual twins, so hopefully this will be an interesting part to finish off with. All right, so the first part is, the Alzheimer's symptoms modeling. So you see here on the left side a number of different symptoms that patients actually experience with Alzheimer's disease. Obviously, at the end of the day, as a therapeutic intervention, you want to actually address those symptoms. The patient doesn't care whether he or she is full of beta amyloid or tau. The patient cares about functioning in daily life. So less confusion, completing familiar tasks, solving problems, poor judgment, trouble with images and spaces, unfounded emotions, a lot of different symptoms there. So the question is, how can we maximize drug effects on the symptoms rather than on the biomarkers? And the idea is the following. There is an approach called computational neuroscience that has been around in academic circles for over 70 years. And the underlying idea is to actually model the neuronal circuits that are expressed in the brain in the appropriate configuration using biophysically realistic changes of action potential dynamics. So here is an example of a cortical microcircuit that we will talk back later quite in some detail, with some of the excitatory pyramidal cells in green and the inhibitory GABAergic cells in yellow, which then couples into basal ganglia, which ultimately we can make models for Parkinson's disease and schizophrenia. But let's focus first on the kind of the cortical macro column here, which is the basis of memory trace performance and memory trace stability. Ultimately, the idea is to use the famous Hodgkin-Huxley equations, which have been, let's say, developed in the late 1940s, to describe the dynamics of opening and closing of voltage-gated ion channels, which then leads to the, let's say, the shape of the action potentials. And we believe that the activity of neuronal circuits in the human brain actually leads to cognitive performance and behavioral performance. A lot of observational studies with both fMRI and intracranial EEG, but also interventional studies with rTMS that suggest that addressing or let's say modulating neuronal firing actually can have behavioral impacts in terms of cognition and other mental states. For the modelers amongst you, just a kind of an intermezzo here about a couple of equations. Basically, the neuronal modeling is based on this particular capacitive current going through a membrane. So you have the capacitive current over here, and then you have a number of currents that actually are related to opening and closing of voltage-gated potassium channel, sodium channels, and maybe a leak channel. And sometimes you have also other types of channels like calcium channels, depending upon the type of the neuron. And so each of those equations actually has a kind of a time-dependent outcome over here that is a kind of empirically fitted to a number of parameters. So it describes very well the dynamics of opening and closing of voltage-gated and ligand-gated ion channels. So once we have those equations, we can then, in a kind of a microcircuit, generate what we call an output, a parameters output, which we call a raster diagram. And in this case, we simulate here as a function of time, which is time in seconds here, the activity of a neural network. And each black dot is an action potential of an individual neuron. So you have different lines here of individual neurons. And so we simulate what we call a working memory paradigm, which should present and sensory stimulus at times equals 2 seconds, and the network actually starts for a very short time. A network starts to fire in synchronization, so in a kind of an attractor state, until, of course, due to the noise, it tends to decrease over here. So we call this a kind of an in silico biomarker. Now, if you talk about pharmacology, we also need to bring in the kind of the drug exposure, and this is done by a front-end module, which simulates the kind of the competition of a small oral molecule, like a dopamine antagonist, the competition of that drug with the neurotransmitter for activating or inhibiting the postsynaptic membrane. So that's necessary for the co-medications, because in clinical practice, many of those patients take a lot of other medications like benzodiazepines, antidepressants, etc. So we need to keep that in mind as well if we want to simulate the outcome of therapeutic interventions on clinical outcomes in clinical trials. Once the activation state has been determined by this model here, we can then couple that by intracellular modeling to changes in effector proteins, mostly the voltage-gated ion channels like potassium, sodium, and calcium channels. And so each of those individual G protein coupled receptors that are activated by those small molecules has their own effect on their own set of voltage-gated ion channels. And we take those data from preclinical studies to populate the model here. The next part is then to bring in the pathology. So the Alzheimer's disease pathology is introduced using from imaging and postmortem data, especially in terms of ApoE-dependent synaptic loss, neuronal loss, and cholinergic state changes, which then are kind of affecting the performance of the network under those conditions. And we calculate the kind of what we call a work memory span, which is the time over which this action potential train can be kept in memory without any further stimulation. So the longer that work memory span, the better you might recognize some of the items that you had in mind before. So it's a measure of functional and cognitive performance. And we have in the next slide, as we can show, a kind of calibration with existing clinical trials. So what we do here is we run a number of historical clinical trials in our in silico model, and then compare the output with the actual observed, let's say, changes in what we call the ADAS-Cog here. And so in this way, we can have a quantitative relationship between the in silico biomarker and the clinical outcome for the patients. Let's now go back to the amyloid aggregation model. So we have set the kind of background for the functional outcomes, and we will come back to that when we bring in the effect of amyloid species, beta species on firing in neural networks. But let's first look at the amyloid aggregation model, which is basically simulating the effect of anti-amyloid antibodies, which have been done over the last 10, 15 years in a lot of clinical trials. So we have a lot of clinical data here, which is a good point, on both biomarkers, amyloid biomarkers, and cognition. But before we go there, one of the key issues with regard to amyloid antibody simulation is modeling the kind of the drug exposure inside the human brain. And so we use for that what we call a mechanistic-based physiology-based pharmacokinetic model, a PBPK model, in which we explicitly model the dynamics of in this case, the antibodies from the plasma into different tissues. So in this case, we lump all the peripheral tissues together, and we focus mostly on the brain here. And we explicitly have a blood-brain barrier, as you can see over here, but also a blood CSF barrier as well, with 4 different CSF compartments, which are anatomically defined. And then we have a kind of interstitial fluids, and of course, then a cellular fraction of inside the brain, which we lump for all the neurons, glia, and glial cells here. And so we have a number of different fluxes and flows that determine the kind of the uptake of antibodies in the human brain. And so we have 3 different processes in which antibodies can actually enter the interstitial fluids. You have the transcytosis over the paracellular pores. We have pinocytosis, or FcR-mediated transcytosis. And then we have also the complex of FcRn with antibodies here. Note that the fraction of antibodies getting into the brain is usually very, very low. We talk about 0.1%, maybe 0.2 or 0.3%. So you lose quite a lot of antibodies if you want to have the mode of action inside the human brain. The next part is then to couple that the dynamics of the exposure as a function of time. Usually those antibodies are given once every month, so an intravenous infusion every month, so you have a kind of a PK profile. Usually the half-life of the antibodies can be between 14 days and 6 weeks. So that's why you have those long intervals there. But so once you have those drug exposure as a function of time, you couple that with what we call the mechanistic model of amyloid aggregation. And here we have different processes. We have synthesis of the A-beta peptide from what we call the amyloid precursor protein. And we have different aggregation cascades. We have, in this case, we have both a shorter A-beta 40 and a longer A-beta 42 cascade, in which monomers are aggregating into oligomers, into protofibrils, ultimately into plaques. And there's a kind of backward and forward rate constant. So here we use the concept of macroscopic kinetic equations to really get to the biophysics of aggregation. So we have primary nucleation, in which two monomers can actually form a dimer, and then another monomer for a trimer. We have secondary nucleation, in which monomers are binding to plaques and have facilitated aggregation with existing oligomers. We have also fragmentation and breakdown as well. And ultimately, we have two different kinds of, at the end, we have also the kind of very important, the clearance of those A-beta species by microglia cells that can go from what we call a resting state, which is the kind of the ramified state that we can see over here, to the kind of hypertrophic state, activated state, which some people call the disease-associated microglia state. And that actually is responsible for clearing the amyloid beta plaques. And they are kind of activated by binding of the antibody to the Fc gamma receptor on those microglia cells. We have two different types of readouts that we can measure from the clinic. We have what we call the SUVR imaging, the standard uptake volume ratio imaging, which is basically a PET imaging approach to monitor the amount of amyloid load inside the living brain, and that we can do that during clinical trial. And you can also look at some of the fluid biomarkers in the CSF, A-beta 42, but also in the plasma, A-beta 42 and A-beta 40 here. Just for the modelers amongst you, this is the equation that we use for instance, for the dynamics of a tetramer. So suppose this is a tetramer here. So we first have a kind of the increase in tetramers from trimers gaining a monomer, which of course forms a tetramer. We have the loss of a tetramer when the tetramer loses the monomer to a trimer with a backward rate constant over here. This is a forward rate constant and a backward rate constant. We have done a forward rate constant of a pentamer gaining a monomer, so one tetramer less. We have done, conversely, a loss of a monomer from a pentamer. We have a trimer gaining a monomer on the plaque with this particular acceleration factor in secondary nucleation over here. We have a tetramer cleared by microglia cells, and we have done quite a lot of effort to really describe the microglia phenotype and the half-life of microglia cells from biological data, so that even in the absence after the treatment of with amyloid antibodies has been terminated, there is still a lingering number of microglia cells that are in the high phagocytosing state. We have also a tetramer losing a monomer on the plaques over here with the forward rate constant. We have the tetramer bound by the antibody. And of course, in that way, the free tetramer is decreased to 1 unit. And we have finally a tetramer moving to a perivascular space as well. So it's really a mass balance equation in which we bring in a lot of different processes to generate the dynamics of individual A-beta species. Now, we can actually reproduce some of the outcomes that have been seen in the clinic, especially in terms of fluid biomarkers. This is an example of a phase 2 study with lecanemab, in which we can pretty well predict or reproduce the kind of the plasma A-beta 42 outcome with the individual's experimental data here, and also the plasma ratio of A-beta 42 over A-beta 40 ratio. Obviously, those are outcomes with a lot of variability there. We can also reproduce the SUVR, the imaging biomarker from the donanemab, which was not used in the building of the model, from the Trailblazer study. So if you bring in the titration schedule of the donanemab, which is first 10 milligrams per kilograms, and then afterwards going to doubling to 20 milligrams, you can see that it can pretty well describe the SUVR dynamics of the Trailblazer study. Similarly, the Graduate phase 3 study, which has a somewhat slower titration phase, as you can see, is also pretty well reproduced by the model. Now, one of the things that we noticed in our development of the model and plugging in some of the properties of different antibodies, because they were, at the time of when we built the model, we had data of about 6 different monoclonal antibodies against beta amyloid, is that lecanemab, for instance, has not much of a difference in terms of the SUVR reduction compared to aducanumab and gantenerumab here. So it's pretty similar. But what differentiates them from the 2 other compounds is the fact that the protofibrils are very much decreased with lecanemab, much more than with aducanumab and gantenerumab. Now, is this the reason why the clinical outcome of the Biogen compound and the gantenerumab compound is kind of a little bit of a shaky outcome? We don't know yet, but I will come back to that later on how much the protofibrils might contribute to the kind of the functional outcome as well. But that's one of the interesting observations in terms of differentiation that the model has generated. The other interesting observation is that we built a kind of an ARIA-E model. ARIA-E is acronym for amyloid-related imaging abnormalities, and basically it's a very nasty side effect that really hinders the uptake of amyloid therapies in the clinical practice, because it's related to microbleeds in the cerebral vasculature based on the activation of macrophages when the antibodies bind to A-beta plaques in the perivascular compartment over here. So we kind of were able to reproduce the observed incidence of that ARIA-E for a number of different antibodies you can see over here, and showed indeed that the aducanumab outcome was much, let's say, a much higher liability for ARIA-E as compared to the lecanemab data over here. In addition, we were able to develop a kind of time-to-event modeling, so to showing indeed that most of the ARIA-E actually happens in the first the first months of the titration of the treatment schedule, and afterwards then to stabilize over here. So now we had, of course, a lot of clinical data from those 7 different antibodies. We have about 32 different observations. So one of the things we were looking at was the plasma phospho-tau readout, because having a PET imaging study is quite expensive, and there is not always PET tracer units in the neighborhood. So I think having a kind of a plasma biomarker for monitoring the kind of the dynamics of central beta amyloid in the brain is very helpful for many patients. So we looked at the correlation between the observed changes in plasma phospho-tau as a kind of an effect size versus the different changes in our model for the plaques, the protofibrils, the oligomers, and the monomers. And what turned out to be the case was that the plaques had a very weak correlation with the change in phospho-tau. And the strongest correlation was with the protofibrils, oligomers, and a little bit more with the monomers. So the more soluble outcomes actually were able to predict the kind of the phospho-tau changes in the peripheral plasma, which is a very accessible biomarker. And if you do a multiple regression analysis, we can actually predict about 70% of the variability. Which leads us to the following point here. So can we use this model to actually help answering questions or challenges in clinical practice? So if the drugs are rolled out in clinical practice, there is neurologist who wants to start his patient on an amyloid antibody. So how can we help them with defining how to run that experiment, that trial? So one of the key aspects is that after a certain amount of amyloid treatment duration, you clear completely the beta amyloid in the brain of those patients. And of course, compounds like donanemab do that very fast, as we have seen before. Other compounds do that somewhat slower, but ultimately all compounds actually, the longer you treat them, the more you clear them. But once the antibody clears, let's say, all the amyloid below a certain threshold, and we call that amyloid negativity, it's time to stop treatment, because you don't want to expose those patients to possible side effects like the ARIA side effects without any beneficial effect anymore, because all amyloid is gone. Obviously, the gold standard for measuring amyloid negativity is a PET scan, is an SUVR scan with the PET imaging tracer, which is, of course, very expensive and not really accessible. So we were looking at some of the peripheral biomarkers for reaching amyloid negativity. What you see here is, as a function of the baseline amyloids, all the way from, we call that centiloids. So usually 25 centiloid is what we call amyloid negativity. And so it goes all the way up to about 200 centiloids. So as a function of your baseline amyloid SUVR, if you start a patient, let's say, at 150 centiloids, your plasma A-beta 42 of A-beta 48 can increase, but it increases with a very specific curve depending upon the nature of the antibody. So lecanemab here actually has the biggest dynamic range compared to aducanumab, and then here is gantenerumab and donanemab. So for each individual antibody, you have to look at a different curve here. Now, a plasma A-beta 42 over A-beta 48 is quite difficult to measure, even if it's a plasma sample, but the assay is quite complex. So looking at the previous slide, we wanted to actually see if we can use maybe plasma phospho-tau changes as a marker for reaching amyloid negativity. Again, we have the same baseline SUVR here, and we can define those curves for donanemab here. This is gantenerumab, this is aducanumab, and here is lecanemab. So we have now a possible way to use plasma phospho-tau biomarkers to define the time at which the amyloid negativity has been reached in the brain of those patients with treatment for the different antibodies. So once you have those amyloid negativity reached, the question is, what is the maintenance dose after stopping the treatment? And here we show the kind of the SUVR dynamically, and you have your cutoff here at 25 centiloids. So everything below this line is what we call amyloid negativity. And so what you see here is the dosing interval in months after stopping treatment. So you can see, for instance, that for donanemab, you can easily go all the way to 64 months and still not reaching the cutoff range over here. But for aducanumab, you reach the kind of the cutoff with a dosing interval between 40 and 50 months. And for lecanemab, it's about 24 months over here. So this gives you an idea on how frequent you should actually treat patients once the amyloid negativity has been reached. The third application that we did with this model was if there is an ARIA-E interruption, if you have that type of nasty side effect, usually you stop treatment for about 3 months, kind of drug holiday, and then you restart treatment. What we did here was we restarted treatment with a very conservative titration schedule for each of the 3 antibodies that you see over here. And we measured the kind of the delay in months to reach amyloid negativity on top of the 3 months. And you can see that the delay ranges between 3 and about 6 months for the different antibodies, depending upon the time at which the ARIA-E happens after a treatment start. So you see here the time in months after treatment start. And finally, we can also do a virtual in silico trial with different patient profiles and actually define a kind of therapeutic index, which is basically the ratio of your biomarker, your amyloid decrease, divided by the ARIA-E liability. And this is the kind of the first step into trying to understand the responder profile of patients. So whether we can identify patients that have the best benefit of risk outcome for each of the 3 different antibodies. Okay, now let's talk about trying to combine the effect of A-beta species dynamics to clinical outcomes. So there is a number of preclinical studies suggesting that monomer of A-beta have quite complex effects on neurophysiology, electrophysiology of neurons. For instance, this is the NMDA conductance, NMDA activity. So you see that the A-beta 42 has a kind of a monotonic decrease over here, but the shorter A-beta forms actually have an inverse U-shaped dose response. They are kind of positive at lower dose, and at higher dose they tend to become negative. And this is the alpha-7 nicotinic receptor, which tend to be monotonic decreasing all over the way. So this is actually published a couple of papers, and it actually can explain 3 different clinical observational data sets on MCI patients, and more importantly, can explain the clinical effects of BACE inhibitors. BACE is an enzyme which we call the beta secretase enzyme, which cleaves the A-beta peptide out of that large amyloid precursor protein. So if you block the BACE enzyme, you reduce the amount of A-beta being produced, and that was, of course, a clinical target for many years. And you could actually reduce the amount of A-beta formation by 80 to 90% using those small molecule BACE inhibitors. But the unexpected finding was that BACE inhibitors did actually worsen the outcome, cognitive outcome, in clinical trials above placebo, which was kind of the opposite of what people actually had in mind. And this is something that could be explained by this particular outcome over here. And the third application of this model is a generate hypothesis for the difference between the lecanemab emerge and engage outcome. Those were 2 different phase 3 trials for the same compound, and they had almost the same effect on beta amyloid reduction, only a small difference between the 2 compounds. But the functional outcome was black and white. So where the emerge trial actually had a positive outcome, so reduced the cognitive deterioration, the other trial actually worsened the cognitive outcome. And we have a kind of a hypothesis for that, which we can discuss if you're interested in offline. Now, the oligomeric A-beta in general increases the excitability. This is the kind of simulation that we did in our computational model, in which we brought in all the different processes that are affected by A-beta oligomer. And one of the things that you can see is that with increasing A-beta oligomer concentration, you have a kind of an increased firing frequency. So the network becomes more excitable when you have more A-beta oligomers present. And this is in line with clinical data, suggesting that amyloid deposition is associated with hotspots in both hyperactivity. There's a number of different clinical data, both in terms of imaging, but also in terms of clinical output. We have what we call subclinical seizures in amyloid patients, certainly in the early stages, so the late MCI, early prodromal Alzheimer cases. Those patients often have subclinical seizure activity based on the hyperactivity due to the amyloid deposition. This all comes together now. So we have this amyloid aggregation model that we discussed earlier, and we can measure the monomer dynamics and the oligomer dynamics. We have that relationship between firing activity and A-beta 42 oligomers. So in the control situation, without any treatment, you have an increased deposition of beta amyloid or increased A-beta oligomer concentration. Therefore, your firing goes up. With an amyloid antibody, you reduces the A-beta oligomer, so your firing goes down, and your network activity goes down. And we know that tau secretion is activity dependent. So More activity means more tau secretion, less activity means less tau secretion, because tau actually tends to hijack the secretion physiology of presynaptic nerve endings. And that's probably the reason why reducing beta amyloid oligomers with amyloid antibodies leads to a decrease in phospho-tau, in this case 217 or 181 as well, which you can pick up in plasma or in CSF. So the change in plasma phospho-tau is quite acute, quite immediate, and is not really related to a kind of change in pathology. It's just a measure of activity of the network, which is affected by the A-beta 42 oligomers. Now, the next step, of course, is the clinical outcome. Can we do a very similar exercise with the clinical outcome? Again, using the 34 observations with the 7 antibodies, and now plotting the change in CDR sum of boxes, which is the clinical outcome, functional outcome, versus the change in our plaques, protofibrils, oligomers, and monomers. Now you see that the plaques become much more important. They account for about 41% of the variants. But also you can see that the soluble protofibrils, oligomers, and monomers also have a kind of an impact there. So the SUVR doesn't tell the whole story, and that's important for those antibodies that are not affecting plaques. There's a number of antibodies in clinical trials that are aimed at oligomers rather than plaques, and they will not have the same reduction in SUVR as, let's say, donanemab, but they can still have a beneficial effect because they affect the monomers in the right way. So if you would do that, sorry about that. So if you do a multiple regression, you can explain about 50% of the CDR sum of boxes. And in that case, both the monomers and the plaques provide a significant, statistically significant contribution to that outcome. Now we have a conundrum. So we know that amyloid antibody treatment reduces firing, as we explained earlier, and you see it over here. This is firing activity. This is the placebo. This is your treatment with lecanemab and donanemab. But how does that explain the fact that the cognitive performance actually would be better with amyloid therapies as compared to placebo? Because we think that more firing is always better behavior, better functioning. Now, it turns out that the human brain encodes information in a very different way, actually more likely to what we call the Shannon entropy. And so one of the ways to illustrate that is if you have a train of action potentials, the red bars over here, you can shift a kind of a green window from left to right and form a number of wards, 4-bit wards like over here. In a very low frequency, you have a certain amount of wards that you can form. When the firing is very high, you have only 2 types of wards that you can form. So there's a kind of a Goldilocks intermediate firing frequency that carries the highest information bandwidth. And a number of recent clinical studies have indeed suggested that the human brain actually uses this type of information encoding much more than just the pure firing frequency. So what happens now is that in reducing the amount of firing, the amyloid antibodies are pushing the system from this particular hyperactivity into this range over here, which actually means more cognitive readout, and that's what you actually see over here. So in our model, what we call the information content actually stays higher with the antibodies as compared to the placebo outcomes. So that's kind of the hypothesis about the reduction in firing on the one hand and the let's say the improvement in cognition compared to the placebo outcome. Let's now switch to the other agent that plays a role in Alzheimer's disease. The next generation of therapeutics will be about tau. Tau is the substrate of the nerve fibrillary tangles. And one of the observations since about 10 years is that tau can actually move from one neuron to the next neuron through a process called transsynaptic spreading. So it's being released on one presynaptic nerve ending because it hijacks the kind of the synaptic physical machinery over here. and brings on its way to the next outcome here is being either pinocytosed or binds to a receptor and is internalized, and in that way jumps from one neuron to the next one, and then starts to oligomerize and form those neurofibrillary tangles in the afferent neuron here. And that's actually, you can see that in the Bragg stages, which is kind of anatomical projection over time with stage 1 and 2, where the pathology starts in the locus ceruleus here, before moving into the hippocampus and ultimately into the association cortices over here. So this is the kind of the process that the tau is moving, the pathology of tau is moving from one neuron to the other. So as a consequence, many companies have tried now to use antibodies to capture that jumping tau from one neuron to the other. So we developed a kind of mechanistic model, again using the same PBPK model for our antibodies. Now we have the release of presynaptic misfolded tau protein and the binding to the postsynaptic neuron over here, and then the capturing of those antibodies of the tau by those antibodies. And so the hope was that by having enough antibodies present in the synaptic cleft, we could actually reduce the amount of misfolded and pathological tau being taken up in this afferent neuron. Now, one of the key challenges is that the synaptic cleft is a very small volume. It's about 20 nanometer width, and an antibody is like 14 nanometer already. So it's a very small area, and 500 nanometer as area over here. It's about 0.04 femtoliter as a volume, so extremely small. So we built this model here using those appropriate volumes over here, and basically this is the equation that we use, which describes the dynamics of the free tau inside synaptic cleft. You have the secretion over here, the source. You have the binding of the tau to the postsynaptic receptor, which is usually what we call an heparan protein sulfate or an LRP1 receptor. We have the unbounding, so the backwards reaction here, sorry. We have the complexation with the antibody, and ultimately the kind of the half-life of the diffusion of the tau out of the synaptic cleft by pure isotropic diffusion. Now, if we apply that model to, in this case, lecanemab, which was a Biogen anti-tau antibody, first we can reproduce the plasma profile quite well. This is the plasma profile of different levels of different doses of lecanemab. We can reproduce the what they call the biomarker. So in the CSF, you can measure the amount of monomeric tau, which is being captured by the antibody. So we can reproduce that quite well. These are the points are experimental data, and then the lines are the simulated data. But the big surprise that when we looked at the dynamics of tau in different compartments, it turns out that the misfolded tau protein in the synaptic cleft is only very limited reduced, only maybe 10% reduced. And even worse, the amount of tau being taken up in the afferent neuron is not reduced at all, which is what you see over here. So the seed-competent tau is building up in the afferent neuron, not different from the placebo, even with very high levels of the antibody. And that might explain the failure of the clinical trials with a number of different anti-tau antibodies, lecanemab, lecanemab, and semorinemab. Now, one of the critiques was that the model using simple differential equations, ordinary differential equations, assumes a kind of steady state mixing in the different compartments, and that there was enough steady staters. But now we're talking about very small volumes in which you have maybe a handful of molecules, maybe 2 or 3 molecules at the same time. On top of that, the synaptic cleft is a very crowded area. As you can see, this is a study from the Physiology at 2015, because there's a lot of proteins that bridge the postsynaptic membrane there, the neurexins and other proteins. So what we did was looking at individual trajectories of individual tau molecules using what we call a spatial Mont-Carlo simulation approach, in which you can actually now simulate the movement of individual particles inside such a volume. So we have this volume over here, which is synaptic cleft, So you can see plus an additional ISF volume that is associated with the synaptic cleft. And you have different interactions. We have the diffusion of tau from the pre to the postsynaptic membrane. We have the binding and unbinding. We have the diffusion of tau from the synaptic cleft inside the ISF and back again. And then we have the antibody binding and unbinding. And so here is different. This is more like a realistic geometry. These are the kind of the nanocolumns that we call the bridges, which is the kind of the proteins that are inside the synaptic cleft over here. So we can then, in this case, simulate individual trajectories. This is an example of simulation in the absence of antibodies. You see that most of the seed-competent tau is being internalized, and only a small number is being released in the ISF over here. Now, when you add antibodies, in this case, non-selective antibodies, because all of the tau antibodies did not make a difference between the monomeric and the seed-competent tau, you see that in the no-bridge case, you might see some effect on the amount of internalization over here. But once you bring in the kind of the nanocolumns, in this case, spaced at 80 nanometer, and in this case, in the green case, spaced at 8 nanometer, you see that there is almost no effect at all of tau antibodies. Moreover, the kind of maximal concentration you can reach with, for instance, 8,000 milligrams of semolizumab is only like 120 nanomole. So we're really talking about this particular area over here. And I think that underscores the reason why we think that lack of sufficient target engagement can explain the clinical trial failures of those first generation antibodies. So once the antibody is binding to the postsynaptic, once the tau is bind to the postsynaptic membrane, we have an uptake process over here. And then, of course, the tau is being processed inside the neuron, is moving with axonal transport from one side into the compartment to the cell soma, and then to the axonal projection, in which it can be secreted to the next neuron over here. And so in the meantime, because the tau spends most of its time inside the neurons, we have a number of different processes that can be, let's say, addressed by therapeutic interventions: oligomerization, degradation, and secretion of synapses. So preliminary studies suggest that we have indeed different outcomes for different interventions. This is if you reduce the amount of the processes by a number of different percents over here, we can see that you can reduce the buildup of the pathological tau over time. Let's finish up with bring it all together and looking at virtual twin models for personalized treatment in cognitive functioning. So in clinical practice, the cognitive performance is very, very noisy. This is just an example of the ADNI database in which we have the ADAS-Cog, the same ADAS-Cog that we talked about, function over time for individual patients. You can see it's like a spaghetti diagram. And so the variability is due to a number of different factors. We have co-medications because those patients take benzodiazepines, standard of care, antipsychotics, antidepressants. On average, about 8 different medications per patient. We have common genotype variants on dopamine and serotonin that affect also clinical outcome. We have the A-beta pathology, we have the tau pathology, and also various comorbidities, vascular pathology, Lewy body disease, alpha-synuclein deposition. So we try to actually mitigate the effect of those first 4 different aspects. We have now an idea about A-beta pathology, tau pathology. We talk about communications and some genotypes. So what we did here was to look at the variability of both the progression rate on ADAS-Cog and the baseline on ADAS-Cog for a large number of virtual patients that had benzodiazepines, antidepressants, cholinergics, antipsychotics, and then with different common variants. We have a serotonin transporter variant, we have a dopamine degradation variant on the COMT enzyme, and we have an ApoE variant. And you can see that if you run the simulation, the progression rate is about 7-fold difference between the lowest, the slowest, and the fastest progressor in this case. And the baseline is also a 14-point difference. And this is just in the absence of amyloid and tau pathology, or the same background, not even including amyloid and tau pathology. So this is a reason why you have so much variability in clinical outcome, why we need 1,000 patients for the clinical trial to really remove that fluctuation and the effects of those co-medications. Now, how do we bring in the tau pathology? We talked about bringing in a beta pathology a couple of slides earlier of a beta monomers and the beta oligomers. How do you bring in the tau pathology? For that, we rely on some of the experimental data with human iPSC cells, in which we had, in this case, a tau mutation or frontotemporal dementia, which predisposes the tau to aggregates, very fast to aggregates. And so when you cultivate those neurons on a multi-electrode array, you can actually measure the kind of the action potential, speed, and dynamics. So what we did was we built a neuron model that was identical to the, well, more or less identical to the experimental neurons over here, with a long axon and cell soma and a dendritic tuft. And then actually changing, the idea was then to look at changes in potassium and sodium channels that could reflect or could align with the observed clinical phenotype. And so it turns out that to get to the changes in action potential duration and speed, the tau had to affect potassium and sodium channels in the what we called axon initial segment, which is the segment right after the cell soma. And there is other evidence as well from other labs that if you inject misfolded tau protein, you can change the potassium and the sodium channel conductances. So now we have a way to actually bring in all those different players into the system. So we have a way to bring in the beta amyloid pathology, the tau pathology in our work memory paradigm system here, in which we calculate either the work memory span or the information content. And so one of the ideas is that if you have a virtual clinical trial with patients in which you have baseline co-medications, of course the genotypes do not change. You have your baseline amyloid loads, you have your tau loads. You build a kind of a virtual patient trial with identical distribution of those different variables with appropriate target exposure and engagement. You run that into the mechanistic model here, and you derive a kind of natural trajectory of the placebo, so the untreated patient over here. And then you do the actual clinical trial with your actual outcome here, and you can now compare on a per patient basis the predicted trajectory of placebo with the observed active treatment of your patient. And that, of course, would maybe hopefully reduce the variability in clinical trials and the need to reduce the trials from, let's say, 1,200 patients could maybe down to 800 patients, which would speed up the approval of certain drugs that might work. This can also be used for rare diseases in which we don't have enough patients to do placebo-controlled trials. So there's a lot of possibilities there. So finishing up, kind of conclusion here. Hopefully, they've shown that this modeling with the experimentally inaccessible intermediate A-beta species, because you cannot measure oligomers and protofibrils inside the living human brain, but they have a biological function. So the next best thing is to actually model the dynamics there. I think a very interesting observation is that you can use plasma phospho-tau changes as an easily accessible biomarker for monitoring what's happening inside the amyloid load into the living patients. We can now bring in the effect of amyloid and tau oligomers, the co-medications, the common genotypes, to kind of mitigate the variability here. And a number of different applications, clinical trial design of amyloid tau. We didn't talk about neuroinflammation, but we also have a kind of neuroinflammation model that links amyloid with the TREM2 receptor. And then very importantly, I think something that many people have not let's say underestimated is the pharmacodynamic interactions with genotypes and co-medications. Clinical practice decision optimal combination of targets. The future directions are understanding why clinical outcome with amyloid antibodies declines with increasing baseline pathologies. So from the large data set we have with the 7 antibodies, it turns out that they work best in the kind of the MCI stage, the prodromal stage, and the very early Alzheimer's stage. But as you go further in the pathology, the clinical outcome is much less clear there. The role of tau pathology in amyloid antibody response, I think, is one of the questions that the FDA is now asking for the lecanemab trial. So that could be a way to look at, because we have the tau pathology outcome for clinical relation to clinical outcomes. And then one of the other things is maybe responders identifications to biomarker changes. So just finishing up with the acknowledgements. These are all the people that have worked on the project, all the way from back in my days with in Silico Biosciences, Aidan Spiros and Patrick Roberts, and then a number of people currently at Certara. So thank you for attending and open for any questions.
Thank you so much for a great presentation. We have a few questions in the chat. Ruchira, would you like to ask your question?
Well, I kind of inferred that the answer was yes, but my question was whether the glymphatic flow was included, that being within the glymphatic vessels being within the recently discovered ones within the brain.
That's a very good question. And so right now in the PBPK model, we don't have any glymphatic vessel there. But I know that there are a few attempts to couple glymphatic clearance of misfolded proteins as well in a kind of mechanistic model. So the short answer is we don't have it for the moment in the model, but it's certainly something that we need to take a look at, because there is increasing evidence that a glymphatic flow actually helps to clear some of the misfolded A beta and tau proteins from the brain. Yeah.
So I was mistaken, but thank you. I appreciate your answer.
Any other question? I see that Shankar may have one. How good is your fit to the coefficient of the model? But you might want to unmute and make the question yourself.
Sure.
Tell us a bit.
Sure. So my question was, because these are mechanistic models, they have a lot of coefficients. And I was wondering, how are they actually, how are these coefficients determined? I would assume by fitting on some.
Yeah, that's a very good question as well. So I think the kind of the calibration of the model, or let's say the parameter fixation, is done in a number of different layers and steps. So this working memory model, that is the kind of the basis for the functional outcome, has been used in a number of other indications as well, including schizophrenia, and not only Alzheimer's disease. So we have a number of different layers in which we gradually have improved the calibration, and ultimately we have many more data than parameters to fit the data. So it's not like an overfitting of the outcome. Now for the amyloid aggregation model, we have also limited our number of different parameters to actually fit to a number of experimental observations. So not only the kind of interventional observations with antibodies, but also the kind of natural history to make sure that the absolute amount of oligomers and protofibrils, which are the kind of the soluble forms, and the insoluble plaques, are kind of in line with experimental data from post-mortem brain tissue as well. So we try to reduce the number of different parameters. So like, for instance, the backward and forward rate constants for the different subspecies. We have experimental data for the first 3 orders. So for the monomer to dimer and dimer to trimer, those are kind of fixed from experimental data. But once you go beyond that, we have a kind of extrapolation using only 2 parameters. to go from all the way to the protofibrils and the plaques there. So that's kind of where we try to limit the number of parameters in order not to overfit the system. But it's certainly a good question that, and the fact that we can now, let's say, reproduce some of the say data sets that have not been used for building the model gives some confidence that it actually is a stable model.
Okay, thank you.
And I think Jane has another one. Jane, you want to unmute yourself? Go ahead and make your question.
Yeah, thank you. Nice presentation. I mean, this is somewhat related to a previous question, since we have so many parameters, and now you have explained that a lot of parameters actually have been fixed, and so you don't calibrate your model to those to clinical data. Then how did you decide those variables to be included in creating VP? My understanding is that if you fix a lot of parameters, then we are not allowing them to identify sensitive parameter. But then in your presentation, it was not clear to me how genotype or co-medication has been included in your model, and then subsequently you will be able to use them as a variable to create VP. Would you kindly explain? Thank you.
Yeah, that's yeah. I didn't go into much detail about how to bring in the co-medications, but that has been the work of the last couple of years. So it's actually based on experimental and clinical data as well. So, for instance, if you have an antipsychotic like risperidone that some of the patients actually take, we know the kind of the affinity profile of that drug for all the different receptors that it affects, like dopamine D2, D1, serotonin 2A, serotonin 1A, and we take that from the PDSP database from North Carolina. So that's the first data sets that kind of fixed. And then we have the kind of the drug exposure, which we actually determined from PET imaging studies in human volunteers or in patients with schizophrenia, based on displacement of a D2 radio tracer by the same antipsychotic. So we get a very good idea about the intrasynaptic concentration of the drug that affects the postsynaptic receptors. And then the physiology of the dopaminergic synapse is also based, in this case, mostly on a combination of preclinical and clinical data as well, to get an idea about how much dopamine is released in natural firing conditions. And then we can calculate the competition of dopamine for the binding site with the antipsychotic on board, and determine the kind of the change in receptor activation state that then leads to a change in the voltage-gated ion channel conductance. And as an example, we had I didn't show that here, but we had a project with a novel Alzheimer drug on serotonin 4 receptor, serotonin 4 partial agonist receptor. So we took the kind of the preclinical data that was very extensively documented in terms of the affinity, the binding of that drug to the receptor, and the amount of activation, and simulated the kind of a phase 1 study in human volunteers with scopolamine, and predicted that the compound would actually worsen the outcome rather than improve it. So the kind of opposite prediction, which of course at the time was a very bold prediction, because I mean, telling the client that your drug is going to worsen it rather than improve it is a very bold prediction. And when they did the trial, the phase 1 trial, actually, and this published in 2013, they saw indeed a worsening when the drug was on board. So suggesting that the model could actually prospectively predict an unexpected outcome that then was confirmed later on in a clinical trial. So I think in terms of co-medications, that is something that we have a good, fairly well confidence in, that we can actually, let's say, bring in the pharmacology and their effect on the clinical outcome for the virtual patient trial. The genotypes, like the COMT and the serotonin transporter genotypes, are a little bit less clear because we have PET imaging data on the effect of that genotype in unmedicated healthy volunteers, which you bring into the model as well. And so yeah, that's how you bring in the kind of the co-medications. I don't know if that's is an answer to your question or.
Thank you. Yes.
Okay.
Very good. We have another one from Jean-Marie. Please go ahead and make your question.
Hello, Hugo. Thank you for this great presentation. I was just curious about ApoE, if you had included that in your model, and if not, if you were thinking of adding this, or.
Yeah, we have ApoE included both in the amyloid aggregation model as a modulator of microglia clearance, again based on the clinical studies with iPSC cells from ApoE4 carriers versus non-ApoE4 carriers, in terms of how much they can actually phagocytose beta amyloid. So the non-ApoE4 carriers have about a 50% higher phagocytosing capacity than the ApoE4 carriers. That's the way how you bring that into the amyloid aggregation model. And we can indeed show that the delay between for aggregation, where we do natural history from birth on to 100 years, we can see that the shift is about 6 to 7 years between the ApoE4 and non-ApoE4 patients. So the non-ApoE4 patients actually get aggregation about 6 to 7 years later than the ApoE4 carriers, which is kind of a little bit in line with the clinical data there. So that's one way how you brought in the ApoE into the aggregation model. In the working memory model, the Alzheimer functional model, we bring ApoE in as a way to reduce or increase the amount of synapses. So we know that ApoE4 carriers have a somewhat lower density of synapses as compared to the non-ApoE4 carriers, and that is the way how you bring that into our neuronal pharmacology model.
Okay. Oh, thank you. Yeah, that's great.
Okay, I don't see any other question in the chat right now. And we are a little bit past our time, but I guess if there is some other question, last minute question that anybody would like to make. Well, if not, I would like to thank again Hugo for coming up today and tonight for him, I guess. And it was very good talk. I'm sorry for missing the very first 10 minutes. And I will close it here. And I hope to see many of you in the next few weeks for our next meeting. Bye-bye.
Excuse me, can I ask a question?
Oh, yeah. A last minute question. Please do.
Yeah, your seminar series is so educational, and I only receive it when IMAG meeting is canceled. And I'm just wondering, how could I get to be notified about your seminars? It seems to me you have bi-weekly, right? But I only receive it every so often. Would you kindly let me know how I can sign up?
Yes, I will share my mail in the chat, and please send me an email. And there might be a chance that your mail gets bounced or something, but I send it regularly every week, and we normally meet every couple of weeks. So,
okay, so you will send me your you put your email address there?
Yes, in the chat, and everybody can get in touch with me if they want to propose speaker of themselves, or if they want to be included in the mailing list. This is a simple one. So please get in touch if you need any information on this side, and obviously get in touch with the lead of the group. So, and you can find every information on the wiki page.
Thank
And
you.
I can share that in the chat as well. So just for you to, you have everything. Give me just a second to retrieve it.
And if you have any questions afterwards, just email me at hugo.geerts@certara.com. So that's
okay. Copy link, and so all info here. And you can also, okay.
Yeah.
So again, a good day and good evening to everybody for those that are on the other side of the ocean. And I'll see you all in a few weeks, I hope. Bye-bye.
Bye.
Thank you.